This is a Professor CBMD PhD, and today's Saturday, October 5th, 2024. It is 9:04 and 13 seconds, p.m. Eastern Standard Time, and today's research, histology methods. So let's hop to it here. Histology microscopic anatomy is a scientific study of microscopic structures of tissues and organs of the body. Light microscopy for viewing glass slides and virtual microscopy for viewing digitized microscopic specimens on a computer screen or mobile device are the most commonly taught methods for examining cells, tissues, and organs in histology courses. Routinely prepared hematoxylin and eosin H&E stained sections of formalin-fixed tissue are the specimens most commonly examined for histologic studies with a light microscope. The first step in preparation of a tissue sample is fixation, which preserves structure and prevents enzymatic degradation. In the second step, the specimen is dehydrated, cleared and then embedded in paraffin or epoxy resins to permit sectioning. In the third step, the specimen is mounted on a glass slide and stained to permit light microscope examination. Specific preparations are required for expansion, microscopy E/M, in which specimens are infiltrated with hydrogels that cause physical expansion of specimens. Steps in specimen preparation for the transmission electron microscope TEM are similar to that for light microscopy, except that they require different fixatives: glutaraldehyde and osmium tetraoxide embedded media, plastin and epoxy resins, and staining dyes, heavy metals. Eosin is an acidic dye, pink, and carries a net negative charge. It reacts with the positively charged cationic groups in cells and tissues, particularly amino groups of proteins eosinophilic structures. Hematoxin acts as a basic dye, blue, and carries a net positive charge. It reacts with negatively charged ionized phosphate groups and nucleic acids, basophilic structures. The periodic acid SCIF-PAS reaction stains carbohydrates and carbohydrate-rich molecules a distinctive magenta color. It is used to demonstrate glycogen in cells, mucus in cells and tissues, the basement membrane, and reticular fibers, and connective tissue. Immunocytochemistry is based on the specificity of the reaction between an antigen and an antibody that is conjugated either to a fluorescent dye for light microscopy or gold particles for electron microscopy. Both direct and indirect immunocytochemical methods are used to locate a target antigen in cells and tissues. Histochemical and cytochemical procedures are based on specific binding of a dye with a particular cell component, exhibiting inherent enzymatic activity. Hybridization is a method of localizing mRNA or DNA by hybridizing the sequence of interest to a complementary strand of a nucleotide probe. Fluorescence in situ hybridization FISH procedure utilizes fluorescent dyes combined with nucleotide probes to visualize multiple probes at the same time. This tissue is used extensively in genetic testing. Autoradiography makes use of a photographic emulsion placed over a tissue section to localize radioactive material within tissues. And this is the Professor of Microbiology, Pathophysiology, Bioinformatics, and Leader of Research. Well, this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day. This is a Professor CBMD PhD, and today's Saturday, October 5th, 2024. It is 9:04.